Hello friends, today we will be seeing Histology of Integumentary System Part 2. In the video on Part 1, we have seen the basic structure of skin, the layers of epidermis and the parts of dermis, and we studied two important slides, thick skin and thin skin. Today we will be seeing the cells present in epidermis and the different appendages of skin. So let us first see the cells of epidermis. There are four types of cells that are seen in epidermis: keratinocytes, melanocytes, Langerhans cells and Merkel cells. Keratinocytes constitute 85% of cells of skin. They are the predominant cell type. Germinal cells of stratum basale divide mitotically and give rise to keratinocytes. Their function is production of keratin and formation of the epidermal water barrier. The deeper layers of cells are basophilic due to presence of rough endoplasmic reticulum that is required for synthesis of keratins. Keratins assemble to form keratin filaments or tonofilaments. Keratin filaments form bundles of tonofibrils in the superficial layers of keratinocytes. These superficial layers do not have nuclei and cell organelles and give eosinophilic appearance. This is a hand-on picture showing us the epidermis of thick skin, and what we see here is the stratum basale, and this here is stratum spinosum. The cells which are seen here, the predominant cell type, are the keratinocytes. We also appreciate the other three layers here, the stratum granulosum, stratum lucidum and the stratum corneum. We then go on to see the melanocytes. These are derived from neural crest cells. They are present in stratum germinativum or stratum basale. They show dendritic-like processes arising from a rounded cell. The processes of melanocytes lie between the keratinocytes. The functions: melanocyte synthesizes melanin and transfers it to the melanin non-producing cells through their cell processes. This melanin transport is an example of cytokine secretion. Melanophores are cells that receive melanin from melanocytes. In the same picture here, in the stratum basale, this rounded cell with cell processes is the melanocyte, and these cell processes are going to be lying in between the keratinocytes. Dendritic cells of Langerhans. These are antigen-presenting cells of epidermis. They are derived from lymphoid precursor cells of the bone marrow. These cells are seen in stratum spinosum of skin, and they are also seen in the oral mucosa, vagina and thymus. They phagocytose bacteria and present bacterial antigens to T lymphocytes. Langerhans cells constitute a part of mononuclear phagocytic system. They are also responsible for delayed type of hypersensitivity reaction. In the same diagram again, what we see here in the stratum spinosum is the Langerhans cell, a large oval cell with a spherical nucleus and with dendritic-like cell processes. So this is the Langerhans cell, dendritic cell of Langerhans. And the last type of cell is the cells of Merkel. These are sensory cells responsible for cutaneous sensations. They are located in the stratum basale. They act as mechanoreceptors and are more abundant in the fingertips, in the oral mucosa and in the hair follicles. The same picture here, in the stratum basale, we see the Merkel cell. We now see the blood supply of skin. Epidermis is avascular and gets its nutrition by diffusion. Skin receives its blood supply through papillary plexus located just below the dermal papillae, reticular plexus located just below the dermis, and deepest plexus that lies over the deep fascia. Let us now study the nerve supply of skin. Skin is supplied by cutaneous nerves that carry sensation from three nerve endings: Meissner's corpuscles, Pacinian corpuscles, and Arrector end organs. Sympathetic stimulation of blood vessels of skin produces vasoconstriction and diverts the blood away from the skin. Autonomic nerves increase the secretions of sweat glands by contraction of myoepithelial cells and also produce contractions of the erectoris pylorum muscles. Now we will study the appendages of skin. Appendages support the skin and they include hair follicle, hair, nail, sebaceous glands, and two types of sweat glands, the Eccrine sweat glands and the Apocrine sweat glands. Hairs. Hair covers the entire body except palm, sole, sides of hand and feet, ventral surface and sides of digits, lips and some part of genitalia. Hair growth and texture are influenced by hormones. Let us see the parts of hair. Shaft. This is the visible part of the hair that projects above the skin surface. Root is the part of hair embedded in the skin and enclosed by the hair follicle. Bulb. This is the lower expanded end of the hair root. Here is a hand-drawn diagram of thin skin and we see the parts of hair here. This here is the hair shaft and this here is the hair root and its lower expanded end is the bulb. We also see the hair papilla and the hair follicle. Hair is a modified part of stratum corneum of epidermis. From within outward, it shows three parts: medulla, cortex, and cuticle. This here is a section passing through the hair, showing the central medulla, surrounded by the cortex and the outermost cuticle. Hair follicle. This is a knob-like structure covering the hair root. It's a dermo-epidermal covering that produces hair. The wall of hair follicle has three layers from within outwards: inner root sheet, the outer root sheet, and outermost dermal connective tissue sheet. This here is a picture showing us the hair follicle which surrounds the hair root, and if we take a section at this level, the cross section appears like this. In the center we see the hair with its three parts: the medulla, the cortex, and the cuticle, and outer to that we see the wall of the hair follicle showing the inner root sheet, the outer root sheet, and the outermost dermal connective tissue sheet. So three parts of the hair and three parts of the hair follicle is what we see in this picture. Another picture, an H&E stain slide showing is the section passing through the hair follicle, showing us the three parts of the hair: the medulla, the cortex, and the cuticle, and three parts of the wall of the hair follicle: the inner root sheet, the outer root sheet, and the dermal connective tissue sheet. This here is an obliquely passing section through the hair follicle. These two are the oblique sections. We then go on to see the Arrector pili muscle. These are small, smooth muscle bundles attached at one end to dermis and at the other end to the connective tissue sheet of the hair follicle. The attachment of the muscle to the hair follicle forms an obtuse angle with the skin surface. These muscles are supplied by sympathetic nerves. This here is the same hand-drawn picture of thin skin, showing us the Arrector Pili muscle attached to the hair follicle at one end and to the surface of the skin, that is the dermis part of the skin, at the other end. What we also see here are the sebaceous glands, which lie here between the Arrector Pili muscle, the skin surface, and the hair follicle on the other side. The actions of the Arrector Pili muscle. Contraction of the muscle produces hair stand on end with slight depression at the dermal attachment of the muscle, giving goose flesh appearance. Sebaceous glands lie between this muscle and the skin surface. Hence, when this muscle contracts, sebaceous glands get squeezed and release the sebum into the hair follicle. We now study the sebaceous glands. These are holocrine, simple saccular glands present in the dermis. They are present in the angle between the Arrector Pili muscle and skin. They usually open into the pilosebaceous canal at the infundibulum of the hair follicle by a short duct. The same picture here showing us the sebaceous gland, the secretory part, and this is the short duct by which they open into the hair follicle. Each sebaceous gland has two parts, secretory part, that is the alveoli, and the conducting part, which is a broad short duct. The duct is lined by stratified squamous epithelium. Each alveolus consists of mass of polyhedral cells and does not have a lumen. It shows two types of cells, basal cells and secretory cells. The basal cells are outermost cells that rest on the basal membrane. They divide mitotically to form the inner secretory cells. Secretory cells resemble multilocular adipocytes. They are large polygonal cells with a big rounded nucleus, and the cytoplasm is filled with lipid droplets. During H&E staining, the lipid droplets get washed out, giving a foamy appearance to the cytoplasm. Holocrine secretion. The secretory cells synthesize and accumulate sebum. During this process, they undergo apoptosis. Entire dead cell debris and lipids get secreted into the duct as the sebum. This picture here shows us the different types of secretion, and this here is holocrine secretion, where the secretions are let out along with the dead cell debris after programmed cell death, that is apoptosis. Now we see the sweat glands. These produce sweat or perspiration. There are two types of sweat glands, eccrine and apocrine. Eccrine or typical or merocrine sweat glands. They are distributed all over the body except lips and external genitalia. Apocrine or atypical sweat glands are present only in the axilla, areola, nipple, skin around the anal opening, and external genitalia. Eccrine sweat glands. These are simple coiled tubular glands distributed all over the body except lips and external genitalia. They are most numerous in palms, soles, forehead, scalp, and axilla. They play major role in regulation of body temperature. Structure. The gland has two parts, duct and body or fundus. The duct is long, runs through the dermis, and opens on the surface of epidermis through a funnel-shaped orifice known as the sweat pore. The duct is lined by stratified cuboidal epithelium. The secretory part or body or fundus is a highly coiled terminal part of the tube. It consists of inner lining epithelium, basal lamina, and surrounding connective tissue of the dermis. The picture here shows us the sweat gland having its two parts, the secretory part which lies deep down in the dermis and the conducting part which is a long duct which opens on the surface of the skin by a funnel-shaped opening called as the sweat hole. The lining epithelium of the secretory part is a single layer of cuboidal cells which are of three types. Clear cells which secrete water and electrolytes. Dark cells which secrete proteins by exocytosis, that's the neurocrine type of secretion. And myoepithelial cells, these are contractile cells located between the secretory cells and the basal lamina, and they help in ejection of secretions. The other type of sweat glands are the apocrine sweat glands. These are simple coiled tubular glands. Each gland has a duct and a body. The duct opens into the hair follicle just above the ducts of sebaceous glands. This picture here shows us the hair follicle, and this here is the apocrine sweat gland having its body or secretory part and the duct. And the duct here, as we see, opens into the hair follicle just above the ducts of the sebaceous glands. What we also see here is an eccrine sweat gland, which we have already studied earlier. The secretory portion of the apocrine sweat gland is lined by simple cuboidal epithelium, myoepithelial cells, and the basal lamina, and is located deep in the dermis, or may be also located at times in the hypodermis. The lumen of the apocrine glands is very wide and stores the glandular secretions. During secretion, a part of the cell is pinched off, and that is apocrine secretion. There is a new concept which states that this gland follows neurocrine secretion, that is secretion by exocytosis. We see the other two types of secretions here. This is neurocrine secretion, where the secretions are stored in the form of vesicles, and these vesicles are seen near the apical portion of the cell, and at the time of letting out the secretions, these vesicles reach the luminal surface. There is disintegration of the cell wall of the vesicle as well as the cell wall of the luminal part of the cell, and the secretions are let out. This is neurocrine type of secretion. What we also see here is the apocrine type of secretion, where secretions are seen in the form of vesicles near the apical part of the cell. The entire apical portion of the cell disintegrates to let out the secretions. Modified apocrine sweat glands. Examples are the ceruminous glands of the external acoustic meatus and ciliary glands of the eyelids. Their function is production of protein-rich secretion, including pheromones. We now go on to study the nails. These provide support to the fingertips. They are modified zones of epidermis. Nails consist of a tough protein called alpha keratin. The structure of the nail. Nail shows following parts. Nail plate or body consists of keratinized cells, shows a free distal end and an embedded proximal end or root. It is continuous with stratum lucidum and stratum corneum of epidermis. Nail bed, a highly vascular connective tissue layer that lies deep to the nail plate and is continuous with stratum basale and stratum spinosum of the epidermis. Germinal matrix. This is the thickened germinative zone at the root of the nail plate, which forms the nail plate. Proximal nail fold. Fold of epidermis that covers the nail root. Lonule. Proximal, soft, half-moon shaped white part of the nail plate that overlies the germinal matrix. Lateral nail folds. Epidermal folds that overlap the sides of the nail. Lateral nail grooves. Grooves between the lateral margin of the nail plate and the lateral nail folds. Aponychium. Extension of base of nail plate or stratum corneum deep to proximal nail fold. Hyponychium. Part of stratum corneum that lies under the free edge of the nail plate. This here is a picture showing us the parts of the nail. This is the distal free edge of the nail plate or the body of the nail, and this here is the nail root. We also see the lodule here, a half moon shaped white part of the nail plate. We also see the aponychium, the proximal nail fold, and the lateral nail folds on either side. Another picture showing us the same. Parts of the nail on one side, that's the free edge, the nail plate, the lodule. On the other side, we see the deeper structures, that's the germinal matrix, the nail bed, and the hyponychium, and the nail groove. Thus, we have studied the cells of epidermis and the appendages of the skin in this video. For further reference, you can read Sontaki Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 150 to 164. Thank you.